Good afternoon. So I would like to give a very clear presentation so that we can avoid any burning questions. But respecting the rushing time, so I would like to give the brief about I'm taking the different angle of the seminar to contribute how not just the plant and pathogen interaction, and how the product is being taken to the market with all this upstream research to the downstream product delivery. So with that, I would like to happily present the improving disease resistance in biofortified crop varieties. So basically, all fortification and biofortification is the two sides of the coin. The purpose is the same. We are addressing the malnutrition or hidden hunger, but the approach is two different. So one is a short term, other one is a long term. The biofortification is a long term as we introduce the genetics that repeat your nutrient generation after generation, just like your disease spectrum. So the biofortified crops are mainly is not in the different cultivation practice. It is also similar to any other crop cultivations. So Harvest Plus is one of the pioneer among the global biofortification initiative. It's run through almost 20 years of the challenging program across the globe, including 150 partners and working on the 40 plus countries. So with that, so how our story is begun, so the group can benefit and student can be more, a tangible research can be done in the futures. So the priority comes the nutrition, as you know, like pathogen list, there is a nutrition list as well. So we target only those 3 nutrients which cause, among the top 10 the death cases as per the World Health Organizations. One is the iron, other one is the zinc, and third one is provitamin A. Well, somebody can ask why not protein? Protein is generally considered. So biofortification is started with the biofortification priority index as we talk about the model today. So those model is a conceptual model is already taken care of 20 years back in some of the program like biofortification, which include the sub-index to mix the major index. What is the sub-index? Is the crop production, is the per capita consumption of the particular crop, staple food, and particular nutrient deficient in the particular geography area. So if you combine all together is so-called biofortification priority index, one can go to the website, Harvest Plus website. You can see the interactive map already built. So this is based on the 13 stable crops, so it's more than 120 countries, which gives the donor, the investor, which gives the idea for the researcher where, which crop and how to invest or do the research. So therefore, I given one example, a selected pearl millet that is highly prioritized for iron content, where the countries as is highly mapped is for India and Sub-Saharan Africa. So the next question come, okay, how much nutrient we are biofortifying? This is that one can assume that biofortification not just adding the nutrient into the crop product, it is a regular consumption of those products, okay, a month together or years together as a part of your stable food that should give a measurable health indicators. So therefore, the biofortification definition is fulfilled. So please correct the definition. It's not just increasing the bioavailable in edible parts of the plant. It's not stopped there. It should be after consumption, an individual should has a reflect as a health indicators. So therefore, this is that slide which indicate how much the daily nutrient of particular crop in a given per capita consumption should be met. For iron crops, you can keep 80% of your nutrition, as particularly iron, can be fulfilled with these biofortified iron crops, whereas the zinc crops, you can up to 70%. Whereas the provitamin A, you will have much higher, is almost 100%. So this is that based on the nutritional evidences created by our nutritionist. So therefore, the farmers grown varieties, you can see the light green, what you can expect the farmer grown varieties 20 years before. And what has been fixed target, the middle bar, is a breeder has to achieve with all the resistance, with all the challenges of the production, has to reach this level of nutrient in a given country with that program. And the green ones, what as a network partnership, the product finally is achieved that level. So 20 years back, this is impossible. As we are discussing now is the modeling as the One Health. And this is a very critical example to set, is everything possible if you unite hands and as well as the optimize and use efficiency of the data. So this is the data-driven approach, which gives you that any of the target crops is mostly everything is 100% achieved in 20 years. Not single organization, as I said, 40 plus countries and 150 plus partners. And many donors is asked where the demand for nutrition in agriculture. If you think the nutrition is come from always other than agriculture. But you see, the staple food we eat every day, even your lunch, the staple food is our maximum quantity of your plate. So that has to provide the adequate nutrition. Therefore, that is not being done because farmers not going to demand nutrition to grow his own variety or her own variety. So if it is a high deficiency rate, it should be the first scenario where we call it as both public and private sector. One can develop the product, all type of the biotic and abiotic stresses, so-called target product profile, TPP. You must be heard many of people from IFCPAR or private sector. That's the blueprint of a variety to be developed in next 5 years. This is the evolving mechanism. So one has to build all the needy mandated traits into the one product portfolio. That's the critical. So therefore, we are doing advocate with many of these donors as well as the research priority and policy setting background. And this should be taken very enough care. It's very the high deficiency. Say for example, India is 50% of the woman is anemic. The question is how supplement going to support in a long term, because agriculture should come to the picture. So therefore, the product profile should have those crops which where is feasible. It should have those traits along with the disease resistance. So I come to the central topic of the seminar. The crop development is taking care of the entire chain of process. It's starting of the discovery in the morning set, and even the afternoon we are discussing a lot of discovery, upstream research that has to be evaluated in the product portfolio and integration of the product pipeline. So which is not exceptional than the regular or traditional plant breeding. In the same way, what we do, we act as, we include as many as discipline as possible in this case, not pathologists, not entomologists alone, not even statistical alone. We also include policymakers, the socioeconomists, we also include the nutritionist. So those two, a champion is going to decide what to breed, what not to breed. Because the breeding of the product is for consumer, not just for farmers. So farmers will focus productivity, but consumer will prefer something else, the quality of the product. So therefore, it is very critical to intervene any of the new traits, just like what is resistant in nutrition, to combine together is a very challenging job. But however, the team has been achieved so far. The goal is not to trade off anything, which is farmer preferred yield or disease resistant, or consumer preferred color or taste. This is a very critical example. That's what I would like to present. So that how we screen is just like any other program. This is one of the example slides I used from ICRISAT when I was working with the team for pearl millet. So, every segregating generation, so after crossing, we used to screen a lot of germplasm and crossing segregating material. So you see almost 40 plus entry out of 90. So almost 50% is qualified, is more than, almost all the pathotype for downy mildew resistant. So the downy mildew is the major devastating disease in pearl millet. Therefore, that is a part of mandatory screening, including the high nutrient dense lines. So this bar indicates how the 5 different pathotypes, it's shown the resistant. You can see all the biofortified lines are given adequate resistance. So what I mean resistant is mostly tolerance. So this is also is done. It is highly possible in downy mildew because it's a 50 years work. People are doing both conventional and molecular mechanism to build the resistant gene pyrimidine. But when it comes to blast, that is not possible. It's more, is evolving very faster than DM, and also is the very latest disease. To control, and we need to have the hotspot location to screen adequate screening. Therefore, the data also reflect all the entries what we have screened, we have less number of resistant. But the same case for any other breeding program is the random data. So similarly, we also use like in a genomic data, how we can avoid the G by interaction because we can't do enough phenotyping with the available resources. So we do identify some of the QTL or SNP markers for blast. You can see the intracrust line, male and female, and also susceptible line. But we also work, is developing a diagnostic markers, which is part of, daily business of the breeding program in any hybrid crops. So these are the diagnostic markers, which include both disease resistant, high iron, and high zinc. So you can have the packages. So that set of traits should be screened all the 1,000 lines in a season. So you will select. So this is the efficiency. So then the question will be asked: are you doing in one location as the disease is spread every 200 kilometers is differently behave as the environment is varies? So you can see the India's almost all the millet grown regions and states has been evaluated, and the data clearly shows that there is an integration of both stress, biotic and abiotic. Millet is always in a marginalized environment, but in the nutrient on top of it, both resistant, both blast as well as downy mildew. This is highly possible when you have the multi-location data. So therefore, we would like to always thank the partnership what we had with the private sector as well as the NORDS team, including all state agriculture university, RICAR institution, who provide, as a voluntary as well as sometimes is the project-based location to test. Otherwise, it is not impossible. Therefore, multi-location testing is very important to take the next level of your upstream research to make the downstream product. And in another case is the beans. So biofortifications work on most 8 to 10 crops. So we are looking after both, all the global south is 40 plus country. And this is one of the data from Kenya. Is the common bean is in any day we call it as mostly the rajma. So this is very, very interesting story again, just like pearl millet, which is high in iron. All the biofortified lines, you can see the level of iron level, which is also shown is a very good multiple, double or triple disease resistant, which has been selected for field condition. Here there is no genomic intervention. What I'm trying to say is that as long as you visibly or consciously select either disease resistant or nutrient dense, is also going to be in your seed. As I said, it's all about playing with the genetics. So this all we need is the new technology to reduce the cycle. There becomes the modeling expert should help us what number of cycle we are going to make more efficient use of resources and also efficient of delivery of the product. So the final one is that we do have the good resistance in a biofortified crop culture. So it is not to worry about. Some of the literature says all the biofortification will maybe trade off the yield or disease susceptible. I don't see any evidences so far. Maybe this house has a lot of pathology experts can bring some of the novel topic to discuss further. So another one example story I would like to highlight here in this seminar, the rust resistant zinc wheat released in Pakistan. Pakistan is also one of the largest wheat growing country and wheat consuming country. And Sindhu region is occupy almost 50% of the wheat area in Pakistan. And this is the one single variety occupy 40% of the area after 3 years after release. This is the biofortified zinc variety, but that is again dangerous. One variety is occupy larger area. But how do you resist? So we are working on the pipeline aspect. But what I mean to say is that it's almost 40 ppm zinc. Is the number one in Pakistan, and in the level of zinc, it's almost 50 to 60% higher micronutrient. It is also resistant to yellow rust. It is also tolerate heat tolerance. So you can harvest. I think a couple of years back, the heat wave is very big in India as well as in Pakistan. The wheat yield is drastically go down, and this is those farmers who grow these varieties had a very happy harvest. So this is very important variety. This is the success story of is combining the most demanding varieties and traits together. And this is the seed production, and India is one of the best example. And as I said, Pakistan, here also we did for zinc wheat and zinc rice and pearl millet, which also largely produced. And this has been consumed by millions of farmers and consumers. And mostly farmers, when I say only on farm, who grow, who eat, not buy from the market. So those calculation is yet to be calculated how much is. Another thing is one of our project in Indonesia. So this is that, and again the disease resistant, you can see the UPP either 6 and 7 and as well as 4, which has been identified for high zinc rice in Indonesia. It's going to be released at the end of this November at the Ministry of Agriculture. And this is that is combining with multiple disease resistant as well as high zinc level. So these are all the partnership based breeding. And what I'm trying to say is that the selection makes a difference in terms of any of the product driven approaches or the project. But what we have done mostly we given the freedom to the partners who being expert, but the product advancement is always happening with the multiple discipline involvement. So this is all expertise should be there to advance. So otherwise we may be biased in selection and advancement of some certain product or certain trait biasness. So this is the better example. In Indonesia also we are following the rice crop. To conclude, as I said, nutrition, how is the highly heritable and transmission one generation to other generation, just like your disease resistant. This is highly possible to combine both traits into a single variety or hybrid. So therefore, this is highly recommended to go ahead. There is no stigma of any ignorance of the trade-off, which has been highlighted, but that's maybe the random. We need to look at the very broad spectrum and large scale value. The second one is the most integration of understanding plant nutrition and how it is really the interaction with physiology, because environment is very critical. So those has to be focused, very host plant resistant aspect. And finally, all we know as both micronutrient and macronutrient play a lot of role in plant nutrition, and we need to understand that's also contribute to the control of plant disease. Certain example, say for example, phytate is although anti-nutrient, it is also involved in the defense mechanism in the plant health. So one has to look at, if I want to increase the bioavailability of good nutrition, I need to reduce the molar ratio of the zinc and phytate. One should play very smartly. Okay, so otherwise we are doing injustice to the plant health system. So therefore it is very important, because most of the phosphorus in cereals stored as a phytate. So it's 80% of the phosphorus stored in cereals is as a phytate. So we can't reduce to the below optimal level. So one should understand the crop optimal level. So therefore such understanding of the genetics and mechanisms is very critical. The challenge is always with the environment. As you said, every 2 kilometers evolve the pathogens, and also the environment is most, especially the soil environment, is very critical. So where we come to that next phase of biofortification 2.0 is the microbiome, is agronomy based biofortification. Is the genetics one that contribute 50%, but remaining 50% contribute to the input based agriculture. Either it should be yield or should be the nutrient or disease resistant. So therefore we need to combine the gains come from both management as well as genetics. So this is very critical area one should focus. And next one is the barriers, always that environmentals is very critical, but we have now tool modeling and genomic tools are available, and that is we are far better, we can handle those challenges. And finally, the limited germplasm is screened. I cannot say is available. It's limited germplasm is so far screened is to identify the multiple traits which has either should be in the germplasm unit or should be the breeding population. One should to critically look for it and select and for their either upstream discovery research, either at the student level or the project level, this is more cautious and one should characterize fully. Finally, what we recommend as part of this conference, as I said to the chairman, you can also write and indicate what to be presented in the proceeding. I think I would ask that it is very important to focus the association between nutrition and disease epidemics. Is it true or not? Has that any expectation if the more nutrition planned will give more nutrition to the pathogen growth or infection? That area one has to explore. That is very critical part. And the second one is the integration of biofortification with the durable resistant. Okay, when I say durable, the pathogen as fast as evolve, we need to evolve as the pipeline of the varieties. With the expert like you, it has to be very critical for adaptation of those new innovative varieties at the farmer field. There we can contribute food system. Then you talk about security, then you talk about One Health. I think this is all I would like to share. If any more question, I would like to happy to answer. Thank you.
Long live India and France friendship.